In this video, we're going to go over the fundamentals of rate and titer. And the goals for this lecture are to understand the basics of rate, including the concept of a specific rate. Understand that to change rate, you can change either the biomass levels in the fermentation or the specific rate. That if you change the biomass levels, you will have an impact on yield. Understand the titer basics. So the titer is a result of the biomass levels, the specific ranks, and length of production. And understand that there can be, however, practical limitations of real-world titers. So we jump into rate. As we've mentioned before, there are three primary metrics used to assess the fermentation of any desired product. Those would be rate, titer, and yield. So for our ABE fermentation example, we have a final titer of 10 grams per liter in 90 hours, which gives us an average production rate of 0.11 gram per liter per hour. But we can then go from these volumetric rates that would happen in a cell culture or a fermentation down to what's happening at the per cell level. And that's the concept of specific productivity or specific rate. So specific productivity or specific rate, which I will use interchangeably, is the volumetric rate of a fermentation process normalized to the biomass level. And basically it's a measure of how fast each cell produces product, right? So mathematically it's the volumetric rate divided by the biomass level, and the units are gram per gram dry cell weight per hour. Other units can be used, gram per OD per hour, gram per cell per hour, but we'll use these in this course. So for our ABE example, during 40 to 60 hours, we hit a maximal biomass or a relatively steady state biomass concentration of around 2.5 optical density units. And if you remember from our other lectures, one absorbance unit at OD 1600 is equivalent to 0.35 grams of biomass per liter. So at 2.5 OD, we would have around 0.875 grams of dry cell weight per liter, right? So that's the concentration of biomass we have here. During this period of time between 40 and 60 hours, we have a pretty constant rate. That's 8 grams minus 5 grams per liter is 3 grams per liter in 20 hours divided by 0.875 gives us a number of 0.175 grams per gram dry cell weight per hour. So that's how we can calculate the specific rate. And how do we improve the volumetric rate or specific rate? So given any product, there are really only two ways to increase the volumetric rate of a fermentation process. So the volumetric rate is equal to the specific rate times the biomass level. And so either you can increase the specific rate, so how fast each cell makes product, or the number of cells, right? Those are the two ways that you have, the two levers you have to increase the volumetric rate of your fermentation. If you're going to increase the biomass level, it's going to be done through fermentation strategies, so media formulations, etc. And if you're going to increase the specific rate, it's either a genetic engineering or metabolic engineering challenge. If you do increase the biomass levels, they will require more feedstock, such as sugar, to be used to grow the biomass, right? So the cells need to have to eat to reproduce. And that may decrease the maximum theoretical yield that you could get in your fermentation. Whereas for any given biomass level, if we hold this constant, increasing the specific rate or specific productivity will enable the same volumetric rate for a lower biomass level, increasing the maximal yield to product. And increasing the specific productivity is a primary challenge of many engineering, biochemical engineering, genetic engineering, and metabolic engineering efforts. So this is just to summarize what we just talked about. So the volumetric rate can either be increased by increasing the specific rate or increasing the biomass level. And that if you choose to increase the biomass level, that is a viable way to increase the volumetric rate in the fermentation. You end up pulling feedstocks away to biomass to increase the biomass level, which means that your maximum yield to your product will go down. Now, since every cell is made of protein, right, and since half of the dry biomass is roughly protein, 50% of the gram dry cell weight, we can calculate the level of protein in a cell culture or fermentation as a function of the biomass level, right? So the protein level is equal to the biomass level times 50%, and then converting to milligrams. We convert to milligrams because many of the units that we'll use when we talk about protein levels are in units of milligrams. Or otherwise, the protein level is 500 times the biomass level, and the units would be milligram per liter. Now we have this cell that has a specific productivity or specific rate, right? And we want to come up with a concept called an ideal whole cell enzyme activity or whole cell enzyme activity, which is basically the amount of activity that we're getting for our particular conversion or our particular synthesis per cell or per amount of protein. So for metabolic engineers, the amount of given enzyme in a functional cell is important, not so much the amount of how good the enzyme may be, although they're both important. So for example, we would have a mixture of all the enzymes in our cell. This red enzyme may be the one that's making our product of interest, right? But it's only a fraction of the total amount of protein. So the whole cell enzyme activity would be defined as the amount of product being made divided by the total amount of protein. So it's how good the cell lysate is, right? Or how good the cell is at making the product. So it's the micromoles of product made per minute per milligram of total protein. And it can be estimated by performing biochemical assays on whole cell lysates without purifying the desired protein. Now, this is different in many ways, although the units are similar, from the concept of a specific activity. So, specific activity of an enzyme is the rate of product formation due to a single purified enzyme. So, these activities are purified with biochemical assays on samples of purified protein. And the units are exactly the same. So, it's the amount of product made by an enzyme per minute per milligram of protein, except in this case it's purified protein. So the denominator here is milligram of purified or the desired enzyme, whereas in the whole cell enzyme activity case it's the micromoles per minute per meg of total protein. Both of these historically have been given the units, right? So 1 unit in a biochemical sense is equal to 1 micromole per minute per milligram. Right? But the unit definition doesn't necessarily clarify what milligram you're talking about, if you're talking about a milligram of total cell protein or a milligram of desired protein. And so historically, those specific activity has been used a little inconsistently. And just keep track of what your denominator is. What protein are you talking about? So there are 2 ways to increase the whole cell enzyme activity, right? So the whole cell enzyme activity, if we oversimplify the problem of specific rate, dictates how good each cell is, right? So it dictates the specific productivity of each cell. So how do we change that, right? When we talked about we can change the volumetric productivity by changing the number of cells or how good each cell is. The whole cell enzyme activity is kind of how good each cell is. And that's a function of the specific activity, how good each protein is, times the protein expression level, right? So where the protein expression level is desired protein or target enzyme of interest divided by the total protein. And so firstly, we can increase the whole cell enzyme activity by increasing the specific activity of each given enzyme. So we can increase how good the enzyme is at making product. And this is usually done by finding a better enzyme or by enzyme engineering. And secondly, we can increase the expression level of the desired enzyme, so the amount of it in the mixture, to increase our whole cell enzyme activity. And both of those will increase the whole cell enzyme activity, and both of those will then increase the specific productivity. It can be very difficult to increase the total protein level in most microbes. So using current molecular biology tools for protein expression, you can kind of expect a reasonable expression level of 1 to 5%. That should be doable. You can try to push your expression level up to 50% of the total protein, but that would be more in a case where you're making the protein as a product. So summarizing what we just said there, just like in volumetric rate, there are 2 ways to improve that. There are 2 ways to improve the whole cell enzyme activity, right? The first would be to make a better enzyme, which would affect the specific activity. The second would to increase the expression. And just to remind you that there are limits on how much you can increase expression. So again, many times key challenges in research and development and biochemical engineering around making better enzymes or finding better enzymes. So those are some of the basics of rate, and we can go to titer, which is conceptually very simple. So the titer is basically the average volumetric rate times the length of the fermentation. So the longer the fermentation is, conceptually, the more product you'll make, right? Gram per liter equals gram per liter per hour times hours. And we know the average volumetric rate is equal to the specific rate times the biomass concentration. So we can basically calculate the titer as a function of the specific rate, the biomass concentration, and the time, right? Now this is conceptually what should happen. Now in reality, there can be things that stop titers from increasing, and these are things primarily a result of toxicity, right? So as you increase a product to some level, that product may be inhibitory to growth of the cell, to the metabolism of the cell. It may inhibit the pathway or the enzymes that are making the product. So there can be situations where there is a limit to the titer that's independent on time.